to talk about, Julie. So without any further ado, Gino Adamson, who's in the New College of Florida, rising sophomore in John Weinstein's laboratory.
Hello, my name is Gino, and I will be presenting to you about repurposing Senicapoc for ischemic stroke. So this is just background knowledge about the epidemiological facts about stroke in the United States, about how it's the fifth leading cause of death in the United States, and it is the first leading cause of long-term disability. It has an annual economic cost of $56.2 billion. And what exactly is a stroke? A stroke is when there's a blockage in a blood vessel that prevents the pathway of blood, and it causes loss of function in the brain and there to be neurological deficits to be present. So what happens during a stroke? This causes the cells to die and the cells to injure within the brain. This is because there's a lack of blood flow, oxygen, glucose, and ATP that will then release these health signals referred to as DAMPs, which are damage-associated molecular patterns that will cause the microglia to respond, and they'll become activated and will cause an inflammatory response. So the treatment options are kind of limited nowadays. There's thrombectomy and there's thrombolytics, where there are few, not everyone qualifies for them, and that they have a critical time window. And then there's Senicapoc and the calcium activated potassium 3.1 channel. So what happens during an episode of ischemia is that there's going to be some calcium that goes into the cell. It's going to bind to the channel, which activates it, and it's going to cause the potassium to leave the cell, which is going to depolarize. But with the introduction of Senicapoc, it goes into the pore, it's going to block it, and it's going to prevent the potassium from leaving the cell and continue the depolarization. So Senicapoc was studied in a phase 3 clinical trial studying sickle cell anemia, and it was found to be safe in humans. And then this is the stroke model, the middle cerebral artery occlusion, where there's going to be a filament that gets inserted to the middle cerebral artery for 60 minutes. So it's going to be removed. And there are two groups, the Senicapoc and the vehicle group. So after the stroke, 12 hours after the stroke, the Senicapoc group is going to be receiving injections every 12 hours for 7 days, which they're going to be given a oil solvent that includes the Senicapoc in it, and the vehicle group is going to be given the injections with just the oil solvent. So after that, the stroke rodents will be studied at the cellular level, the chemical level, and at the behavioral level, 7 to 37 days post the stroke. And the brains will be fixed and removed and will be cryoprotected and different molar concentrations of sucrose. So cryosectioning, here's me at the cryostat. Too bad I didn't add the one with me, Madeline, in it. But 30 micron sections were sections started when the corpus callosum is present, and it was stopped when it starts to end and separate. So whenever I took the cryosections, I placed the tissue in PBS room with the optimal cutting temperature compound, and I placed it in a 24 well plate with PBS A-side to promote long-term storage. So then we get into the fun stuff, immunohistochemistry, where the tissue is placed in a block with normal donkey serum and Triton to where open up the cells and prevent from non-specific binding. So what happened was an antibody specific to a certain protein would be binded to the tissue, to the protein of choice, and it would be incubated for 24 hours, and then there would be a wash, and then the primary with the fluorescent, a fluorophore attached to it would also be added to the tissue where it would incubate for 24 hours. And then there would be another wash, and then DAPI would be added to it. And then after that, it would go underneath the fluorescent microscope where it would be studied and analyzed. But the antibodies of choice were NUN, which stands for neuronal nuclei, IBA1, which stands for microglia, and GFAP, which stains for astrocytes. And then DAPI is not a antibody, but it was used to stain DNA. So here's the image analysis. On the right-hand side, you can see a new and a DAPI stain. So each one of these images were inserted into image J, where the right and left hemispheres and the right and left ventricles and the infarct area were all measured. So this was possible by placing different dots all over the image, and it made it possible to make all these different calculations. So, and the picture right there, the yellow, you can see that there's a lack of purple staining, which is nuant, which represents neuronal death. And then in the graph, I mean, not the graph, but in the equation, factored in the possibility that there's edema, or known as swelling, in the early stages post-stroke. And then it also factors in the later stages of the stroke around, normally towards the end, around day 37, where there's necrosis and that there's a bunch of shrinkage. So in the graph, you can see on the x-axis, the different coordinates of the brain, starting towards the front of the brain, and then it goes all the way to the across the back of the brain. And you can see that the Senicapoc had a decrease in infarct area compared to the vehicle group. And then this is another image, which is new in a GFAP, which stands for neurons and astrocytes. And the rectangle you see on the left, this is basically a zoomed in version of that, where you can see the lack of the gray staining. So there's no nuant present, so there is neuron death in that area. You can see how the astrocytes are clustered around the area of infarct. And then this is another image which stands for IBA1 and DAPI, which basically stands for DNA and microglia. And this is a zoomed in version where you can see how all the microglia, how there's the majority of activating microglia that promote inflammation. So this is a summary of what I've learned throughout this summer, is that stroke pathology influenced microglia and astrocytes, and that Senicapoc did decrease the infarct area. And then what could happen in the future is to continue image analysis, compare IBA1 and GFAP staining between the Senicapoc and the vehicle group, and to focus primarily on infarct volume instead of infarct area. So here's a bunch of acknowledgements. I would like to thank Dr. Alan Bogan to make this possible, and everyone else that contributes to the program. I would like to also thank my lab and my mentors for helping me throughout this process.